we take the functional aspect of the Golgi apparatus. When you take the function of the Golgi body, proteins are received from the endoplasmic reticulum. Now these proteins has to be transported. So how the proteins are exactly transported to the destined part of the cell or to the destined tissue in the body? When you take the protein word, proteins are usually called polypeptides. Have you heard this word polypeptides?
I've heard but don't know the meaning.
No problem. So poly always stands for what? Many or numerous. Peptide. Peptide is a bond. Or you can say linkage. Many molecules will be linked together and forms proteins. So what are those many molecules? Those many molecules are amino acids. It's a chain format of reaction happens actually in the synthesis of protein. What really happens is, assume that this is one amino acid and this is another amino acid. Likewise, you will be having so many amino acids. These amino acids will get through diet. Okay, they are important source of protein. Will get through diet. So these amino acids will be linked with each other. Something like this. In some format. This linkage has to be done in a format so that a polypeptide will be formed, which we call as protein. This whole polypeptide, based on its structure, will go to specific part of the transported area. Suppose assume you are having a digestive enzyme. I'm taking the digestive system. It will be convenient for you to understand. That's why I'm taking digestive enzyme. Assume you are having a protease enzyme. I hope you people know what does mean by protease. Yes? No? Protease is the digestive enzyme which will digest proteins. So here you want the protease enzyme to digest proteins. So this enzyme definitely will be in the form of protein. The protein has to be transported towards the digestive system. The digestive system is the destination of this protein. So in order to reach there, these proteins has to be folded properly, and they have to be packed properly so that they will be destined to their particular site of action. So randomly, if we have structures like this, a branched one or radial one, they won't go in a proper direction where they have to act. So to get into the target point, if you assume the digestive system as a target point, the proteins has to be properly folded and uniformly packed for the transport. So this is how the Golgi apparatus will do inside the cisternae tubules. What happens is the proteins which are coming from the endoplasmic reticulum, they will be properly folded and packed, and they will be transported in the form of vacuoles to the destination or to the targeted point. Clear? How the proteins are getting folded and packed is of question, isn't it? How they are getting folded? and how they are packed. That's the functional aspect related to Golgi apparatus as the second point. So first point is always transport. Okay. Even in endoplasmic reticulum also the first function is transport. Transport from endoplasmic reticulum to Golgi. Now even in Golgi complex also the first function is transport. Transport to destination or the target place. Now before transport they are getting modified inside the Golgi apparatus like this. Folding and packaging. How that happens? That is the second function. So here if you take the folding and packaging words, with Golgi apparatus function, here some processing happens. What is the processing? Assume that the same example I'm taking, some digestive enzyme has to be transported to digestive system for digestion purpose. Digestion of any molecule. Digestion of lipid or digestion of protein, any molecule. So this has to go here only, this enzyme. Assume that this is a T enzyme. Just for namesake I'm taking T, transport enzyme. This has to be transported from the site of production, means over here to here. Okay. During the processing of this enzyme, what will happen is the enzyme will be added either a carbohydrate or a lipid to this protein. Enzyme we are saying the protein, basically we are discussing about protein only. Processing of protein which happens in Golgi apparatus is some tails will be added to this enzyme. Suppose assume that this is a T enzyme. Some tail will be added. This tail is nothing but maybe carbohydrate or maybe lipid. This tail part adding to this enzyme is nothing but processing, folding and packaging. All this terminology related to transport of the enzyme from the production site towards the transport site. This is what happens inside the Golgi apparatus. If this tail is not added, the enzyme may go to somewhere else, not to the digestion process. So a tag, you can say a tag is added to the protein, which we call as processing. That includes folding and packaging. Here with these three terms. So here I use the word tail to indicate tag, so that you can understand in a better way. See, suppose I'll just compare a journey. Just in a common language, I'm comparing the whole process with a journey. Suppose a person A is there. Listen carefully, everyone. Suppose a person A is there. He just want to go from Delhi to Goa. Assume, for time being assume. A person A wants to go from Delhi to Goa. He may take the air transport or rail transport, something he'll take, some transport he'll take, isn't it? Some means of transport he'll take to start from this point and to reach here. For the transport purpose, he has to get a ticket, isn't it? If he goes by airlines, he get a ticket. If he goes by railways, he has to get a rail ticket, isn't it? If he takes ticket from Delhi to Goa only, he will reach from Delhi to Goa. Suppose if he takes ticket from Delhi to Hyderabad, assume, can he reach Goa?
No, ma'am.
No. That's what what really happens with the protein here? Assume that A is a protein. Just for general purpose, I'm giving this example for you to understand the terminology in biology. Okay, so we are taking a protein A here. We are assuming the protein A has to get a tag, or in other words, we are saying tail. Any of that we can use. Tag or tail, just this indicates the ticket. Okay, so how this tag gets added to the protein? By processing. So the processing involves either addition of carbohydrates or addition of lipid, something like giving ticket to the protein. Understood? So that the protein will reach its destination. The target will be reached by the protein. Likewise, if you take a ticket to a particular place, you will reach there. Otherwise, you can't reach a place. So that tag or that tail adding to a protein is called processing. Processing of protein means this example you can compare. Forget this. Remember that proteins will be added tags in the form of carbohydrates or lipids, so that they will go to the target place only, not to some other place, which we called as folding, packaging, and processing. So all this happens inside Golgi complex. That's the major function of Golgi complex or the Golgi apparatus. Clear with Golgi apparatus?